Today we are going to talk a little bit about carbohydrates. So let's get started. So first of all, we love carbohydrates. I'm sure all of you have that sweet tang that you look for when you're studying and you just need that sugar rush to get you going, as well as other things like energy storage, and just overall you eat carbohydrates to make yourself a little happy and a little full. But carbohydrates are made of these components called monosaccharides. They're monomers, so monosaccharides are monomers of carbohydrates, and they generally have this formula over here. So we have a certain number of carbons and then H2O, and hence that's why we call it carbohydrate, right? And N is generally greater than or equal to 3. So if we're talking about glucose, for instance, we have 6 carbons, so it's C6. H2 times 6 is 12, so H12, and then O6. So C6H12O6, that's glucose. And you can also have other types of carbohydrates with multiple numbers of carbons. But carbohydrates are very important because they serve a few key functions. The first one is energy storage, more or less short-term energy storage, but nonetheless energy storage. For most organisms, like for instance in humans, it's stored as glycogen. So this is more for animals. And in humans, we can actually store various glucoses together and make a long chain called glycogen, and then we can break down that glycogen into glucose components and then further break down that glucose and ultimately make things like ATP in the process. It can be used also, carbohydrates can be used to store or transport energy. It can act as skeletons for other molecules, and it can also be used for structure. So chitin is this component of exoskeleton of various arthropods, like for instance in insects or spiders and arachnids. And so it has a structural component too in regards to its function. Now, when it comes to carbohydrates, generally speaking, we have a little bit in regards to nomenclature. So first of all, carbohydrate means sugar, and then that can also be sometimes considered as saccharides. So these terms are often interchangeable in many instances, or at least to greater or lesser degrees. But if we break down the individual nomenclature, we can see that monosaccharide is the simple sugar because it's just one single monomer. You have disaccharide, which is two simple sugars linked together by a covalent bond, and we will talk a little bit about that bond in a bit. Oligosaccharides are generally sort of small saccharides where you have 3 to 20 monosaccharides linked, and if you have polysaccharides, it's often hundreds to thousands of monosaccharides joined together. So monosaccharide, for instance, would be something like glucose. Disaccharide would be something like sucrose, and then if you link a bunch of different sugars together, you can make a long polysaccharide, and that's what things like starch or glycogen are. So there you go, some basic nomenclature. Now, when you move on from just monosaccharides themselves, you start seeing some patterns within the whole idea of this carbohydrate thing. And one thing that you might notice is that one of the most common carbohydrates, or one of the most common monosaccharides, I guess I should say, is glucose. Glucose is very interesting because it can appear in sort of a straight line chain, or it can form a ring. And so here this is a ring form of glucose, a pyranose ring. And the ring form is actually more stable. So it's preferred in biological systems due to that stability. In the ring structure, glucose kind of has two conformations. You have alpha conformation or beta, and we will talk a little bit more in the next slide, but this is how you can determine whether it's alpha and beta, and that's by looking at the anomeric carbon over there. So it's looking at carbon 1 of the ring and the orientation, and in biological systems, again, the beta form is somewhat more preferred, but you can find both forms within the body. The reason why the beta form is preferred is because of how everything is structured within this ring. So if you look at the ring here, this is an alpha D glucose. You can see that the OH group, or the hydroxyl group, is pointed down. When it's pointed down, this is the alpha form, and when it's pointed up, it's the beta form. You can actually convert this into this via hand, but you, obviously for the scope of this course you don't have to, but maybe for a biochemistry course for sure you have to learn how to convert a linear form into a cyclic form. But for now, just know that in terms of the carbon 1 or the anomeric carbon, you can see that in an alpha form it's down, the OH is pointed down, and in the beta form it's pointed up. Now, this is very interesting because you can see if you've taken something like organic chemistry that if the OH is pointed down in the alpha form, it's going to have some kind of an interaction with this OH over here in carbon 2. So that's sort of a steric hindrance, whereas something like this doesn't really happen. So this is the reason why the beta form is more preferred. So again, you can look at this instance over here where beta form isn't actually going to have any sort of hindrance. So in the beta form, the hydroxyl groups of carbon 1 and 2 are farther away, and they're not pointed towards each other, and therefore there's less strain. So there you go. That's more of a trivial point, but if that gets asked, now you know. Now when it comes to monosaccharides, they can have a different number of carbons. So you can have 3 carbons, which are what you consider your glyceraldehydes to be. So your glyceraldehydes have 3 carbons. When you have 5 carbon molecules, they're called pentoses, and these include ribose or deoxyribose, which are components of RNA and DNA. And then you can have 6 carbon sugars, which are called hexoses, and these include things like glucose, galactose mannose is another one, as well as fructose. Now when it comes to monosaccharides, you can actually join them together. So monosaccharides are joined together by these things called glycosidic bonds or glycosidic linkages. They're covalent bonds, and they form disaccharides, and then eventually you can add more of them and more of them to make polysaccharides. So they're joined together by glycosidic linkages to form disaccharides, oligosaccharides, and polysaccharides. That's cool, but these glycosidic linkages are very interesting because they're formed through condensation or dehydration reactions. They're also known as dehydration synthesis reactions. So that's very similar to how we form amino acids. So you lose a water and you join them together, and you do that again and again and again until you make a nice long chain, or however many you need to join together. Now, these glycosidic linkages can also be in alpha or beta form. So if you have an alpha glycosidic linkage over here, it's actually obviously the O group pointed down, right? And then with the beta glycosidic bond, the O is actually pointed up. And this is just how it's used to kind of show a beta glycosidic linkage in this instance, and this is how they show an alpha glycosidic linkages. So if you look at the different types of sugars out there, different types of disaccharides, you can see that there are different ways that they're actually going to be bound together, and same thing happens between polysaccharides as well. So there you go. So in alpha, remember, the OH group on the carbon 1 is opposite of this group, so it's often pointed down. And on the beta carbon, in the beta form, then the OH group on the first carbon will be on the same side as the CH2OH over here. So this is another way that you can use. This is actually the better way. If you look at how it's oriented in comparison to this CH2OH group over here, you can actually figure out whether it's beta or alpha. But generally speaking, in the way that it's drawn, often it's nice. And so you can just look at the point up or point down thing, but this is actually more accurate. So if you want to have the more accurate way of determining, use this. So there you go. Okay, so let's move on. So again, when you look at various methods of joining monosaccharides through that linkage, the whole concept is about the same. So you have this type of formation here where you're trying to link two different types of monosaccharides together. So in this case, you're joining a glucose with a fructose. In this case, you're joining a glucose with another glucose. So you have a pyranose ring, a furanose ring. That's also a little bit trivial, but obviously that the key thing is shown here, where through formation of that glycosidic linkage, it requires a dehydration reaction. Different orientations perhaps of how the glycosidic linkages are formed, but in terms of the formation itself, the process of dehydration is relatively similar. Okay, so moving on, if you look at oligosaccharides, sometimes they can actually consider other functional groups. And oftentimes you can see that there are sugars that are sometimes covalently bonded to other types of macromolecules like proteins. And this covalent linkage to proteins and also sometimes lipids can be found in places like the cellular membrane. And this is actually how we get our different blood types through something like this. So depending on the particular sugar, you can actually have linkages to other types of macromolecules, and that can cause variations in the types of structures, as well as variation perhaps in the types of signaling. Once you have all those monosaccharides joined together, you can form a nice long chain, and that's called a polysaccharide. So polysaccharides are just large polymers of monosaccharides, and there are many different types of polysaccharides. There are three major groups that I think we discussed in most of our introductory lectures. And the first one is starch, and this is the type of storage for glucose in plants is through starch over here. So there are actually branched polysaccharides. They have alpha linkages. There are actually two different types of polysaccharide within starch. I think it's amylopectin and amylose, and they're slightly different in their way, but collectively we call them starches. Then there's glycogen, which is the storage form in animals like us, and they're actually also branched polysaccharides, and they have alpha linkages. And then finally we have cellulose, which is a structural component in plants, and it's made of non-branches. So here you go, you can see that they're quite linear and not very branched, whereas in starches and glycogen you can definitely see some branches over there, so see some of those alpha 1,6 linkages. But in cellulose we actually don't see any branching, and it's just a straight chain. The side note and piece of information to that is when you have a lot of branches, it actually makes it easier to be degraded, whereas if it's just a straight linear long chain, it's actually much more difficult to degrade. And the other thing is that beta linkages, I believe we as humans don't actually have any enzymes that can successfully break apart beta linkages. So that's why we can't really digest cellulose. But again, here are the three major groups of polysaccharides that you're most likely going to run into in an intro classroom, as well as probably on an exam. Finally, we can talk a little bit about carbohydrate modification. So carbohydrates, you can stick them together, little monomers together to make polysaccharides, but you can also add in some modifications to them. And one of the things you can do is actually throw in a phosphate group. And by adding in a phosphate group, it can be useful for sort of control, cellular response, and sometimes proteins, when you phosphorylate them, you can make them more active or less active, and it's very critical in controlling various pathways. You can throw in amino groups and attach amino groups to them, and you can form amino sugars as a result, as we discussed earlier. And you can also add in an N-acetyl group, which of course can be useful in some instances for instance, structure. And so in chitin, for instance, in the hard exoskeleton of insects, it can be useful for that structural role. So I think that's basically all there is to the introduction and the basics of carbohydrates. Obviously, carbohydrates are very important to us as humans, as well as to our wonderful planet. So once again, I hope you enjoyed this short little video tutorial, and as always, best of luck studying and good luck on the exam, everyone.